So how do enzymes work? What the mechanism of working? What we have already noted is that the active sites have particular shapes where enzymes the substrates can fit, and then forming that temporary transition state, which lowers the activation energy, and then you have products coming out. Okay, so we have two mechanisms. We call them, one is lock and key hypothesis, and then the induced fit hypothesis. So let's look at each of these and understand how they work. With the lock and key hypothesis, the enzyme is looked as the lock. And for the lock to open, it needs a specific key that has a specific shape. So only substrates that can fit well into the active site can be acted upon, and this explains why enzymes are specific towards their substrates. Okay, so you have the key being the substrate, the lock being the enzyme. Okay, so the key has to neatly fit into the lock for the enzyme to work. And then the induced fit theory is about the fact that enzymes are not really that rigid, but they are to some extent flexible, and this can allow substrate to fit neatly and then form that complex. And so when, for example, the substrate fits into the enzyme, when you have the enzyme substrate complex, the shape is not going to be, the shape of the enzyme is not going to be the same as compared to the shape when the substrate has not binded. Okay, so that is an important point. And when the products have left, you'll find out that the enzyme regains its original shape and structure. So this is what is happening here. You can see that you have an enzyme there and the substrate. Okay, so the substrate enters into the active site, and then you form the enzyme substrate complex. When you look at this structure, it is not entirely like this. There is some conformation that has happened, some transformation in terms of shape that has happened. Okay, and then when you have this, the products have gone, this one now looks like this, but this and this are actually different from that, or different from that. So that is the induced fit theory, which explains that enzymes are actually flexible, and when substrates bind, the enzyme changes its shape, enhancing the ability to bind and facilitate the reaction. And this can be seen in the kinase enzyme that undergoes conformational change to bring ATP and glucose together. Apoenzyme. This is basically, these are enzymes that are inactive forms of enzymes. We call them apoenzyme. They require other compounds or other ions for them to actually work fully. Okay, so they need what we call cofactors, and the cofactors can be inorganic ions or can be organic compounds that they need for these enzymes to work. So apoenzymes are inactive forms of the enzyme that require cofactors for them to work fully. Okay, so this is the inactive one, so it needs another thing that is a cofactor. The non-protein component, it can be inorganic, can be organic, and then when it is there, then you can have it combined with the substrate and then reaction happening. So the whole thing, where you have the cofactor and then the enzyme forming, you have what you call the halo enzyme, the whole enzyme. Okay, so these cofactors, generally we use cofactors to talk about the inorganic and the the organic compounds that are needed for the enzyme to work very well. So in some texts, you see that cofactors basically refer to the inorganic ions. It can be iron 2, it can be magnesium, it can be zinc, but these are very important for the enzyme to work. And then when we talk about coenzyme, now that is when we talk about the organic molecules that are needed for the enzyme to carry out their functions. Okay, so NADH is one of the examples, and then we also have fat is one of the examples of coenzymes. We have the prophetic groups. These are non-amino acid components that tightly bound to the protein and are essential for their function. So, for example, hemoglobin has iron, and that iron, or the heme, the heme in the hemoglobin is what we call the prophetic group.